Greetings, students. So, in our last couple screencasts, we were talking about the Hardy-Weinberg equation as something that describes the allele frequencies in a non-evolving population. And so we define that non-evolving population as one in which the gene frequencies do not change from generation to generation. So if 45% of the genes in a population are big A and 55% are little a, those percentages or frequencies in this case stay consistent from generation to generation to generation. And so if we are at Hardy-Weinberg, then we can use these two equations to do some calculating. And we did. We went and solved some various problems, and the last two screencasts kind of focused on these mathematical aspects. Today we're going to look more at the conceptual aspect of the Hardy-Weinberg equation. In order for a population to be at Hardy-Weinberg, 5 assumptions must be met. And you definitely want to write these down and understand these 5. So to be at Hardy-Weinberg, the following 5 conditions must be met. Number 1, no natural selection. Number 2, no mutations. Number 3, isolation from other populations. Number 4, random mating, none of this choosing partners business. And then number 5, a very large population size. So if all 5 of these assumptions are met, then the population will be at Hardy-Weinberg and the gene frequencies will not change. And that's kind of what this complex figure is showing. Basically, we start with 80% big A or dominant allele, 20% recessive allele, and then after one generation, 80% of our eggs have big A, 20% of our eggs and sperm have little a, and then they come together and form a population where we're 80% big A, 20% little a. And that just kind of keeps going forever, which basically just another way of saying that gene frequencies don't change. So we need all 5 of those assumptions to be met. How often, take a moment to reflect, do you think these assumptions are met? And if you look at those, hopefully you're thinking to yourself, almost never. And that's probably true. Like, for example, we know mutation just kind of happens. And so really, we're always going to be deviating a little bit from this Hardy-Weinberg equation, which means we are always going to be evolving. And that's a pretty monumental thing to say. In order to not evolve, all these things must be true, and it's pretty hard for all those things to be true. So basically, life is continually changing, life is continually evolving over generations. Wow, okay. So what we're going to do now is talk about specific mechanisms of evolution. And basically, any deviation from the above assumptions are going to be causing a change in gene frequencies and therefore evolution. And so any deviation from those assumptions is therefore going to be a mechanism of evolution, a way in which evolution occurs. So our first assumption was no natural selection. And so our mechanism of evolution is going to be natural selection. And this one, of course, was the one that Darwin sort of hit upon. And we've already discussed this in our last chapter. And so you, I'm hoping, have a pretty good grasp of what natural selection means. But again, this is only one of 5 mechanisms that we're going to discuss today. So a little bit more on natural selection that I just kind of want to point out. First of all, remind me, who has greater fitness? An animal that has 1,000 offspring lives 10 minutes or dies, or an animal that has 10 offspring lives 1,000 years and then dies? Hopefully you said the animal with 10 or with 1,000 offspring, because fitness in an evolutionary sense is based on how many offspring you manage to leave behind. It doesn't matter how long you live. What matters is how many offspring you pass your genes onto. That is what is going to make sure your genetic material spreads. So there's 3 types of natural selection. And, you can imagine if you have a frequency of individuals, in this case, we have mouse fur color here. And the frequency of individuals follows kind of this bell curve where most are kind of this brownish color. You got a few on one extreme with the white fur, a few on this extreme with the black fur. If that's your original population, well, directional selection would favor one extreme over the other. So the solid line is going to be our original population. Directional selection would be if, let's suppose, the darker colored mice had an advantage. Maybe the rocks are darker in color or something like that. If that's the case, then we're going to select for more of this phenotype, and the population overall is going to shift. We call that directional selection. Disruptive is where we favor the extremes, but not the intermediate. So maybe there's some really light colored rocks and some really dark colored rocks, but not so many of the intermediate colored rocks. Well, that's going to be disruptive. So we're going to select for the very light colors and the very dark colors. Stabilizing is where we favor the intermediates. Maybe all the rocks are brown in color, and so these mice camouflage in there, have the advantage, and so the extremes are going to be selected against, and we're going to see more and more of this intermediate. So we have these three types of selection. Let's take a peek at some examples of this. So I'm going to give you an example. You tell me which of these three types, directional, disruptive, or stabilizing, this is. Human birth weight. Babies that are too small may die. Babies that are too large may not fit through the birth canal. So which of these three? So we're selecting against babies that are too small and babies that are too large. And hopefully you said this is an example of stabilizing selection, because you can see that this is actual data, by the way. In blue, we have the percent of births in the population, and we see a very nice bell graph in blue. And then we have percent of infant mortality, which is a little sad, but you can see there's more infant mortality in the very small and in the very large based on this data. So this would be an example of stabilizing selection. The size of many crops has increased over time due to artificial selection. So here's some corn. This is the ancestor to corn. Doesn't look all that appetizing, and things have gotten larger over time. Which of these is it? Ding! Hopefully you said directional selection, because we're favoring the larger phenotype. Here's another example. There are large seeds and small seeds available on an island. Birds with larger beaks do better at opening large seeds. Birds with smaller beaks do better at opening smaller seeds. Which graph represents that type of selection? Ding! Hopefully you said disruptive selection, because we're going to favor the small beak and we're going to favor the large beak. Intermediate beaks don't handle either type of seed quite as well. So these are just some examples of natural selection. Okay, our second mechanism of evolution is mutation. So our assumption to maintain Hardy-Weinberg equilibrium is no mutation. Our mechanism of evolution is mutation. Mutation, of course, creates new alleles and eliminates others, is the ultimate source of all genetic diversity. We've talked a lot about mutations in our genetics unit. We've seen different point mutations that can happen, a silent mutation, a missense, a nonsense mutation, a frame shift mutation. We've looked at chromosomal abnormalities with insertions and deletions and translocations and inversions. And so, yeah, so mutations of any kind is going to change the genetic material and therefore cause evolution, change gene frequencies. So some examples of this and how this could play out. One is duplication. So there are some examples in nature where we've seen that a gene has a duplication, and we've seen how this can happen through a chromosomal duplication, for example. But if you have a duplication, as happened here, so we got a gene with 4 different functions, but then it's duplicated, what happens is, since you already have those 4 genes doing what they need doing, that leaves these 4 to start to just kind of change, and maybe they take on a totally different function. And so there's a very interesting example of this with the antifreeze gene in ice fish. So this is a really good video clip. I strongly recommend you click that and watch it. But basically, these fish are like transparent, and they live in just crazy cold water. And what they found is that there was a gene for a pancreatic enzyme, and that gene duplicated. One copy continued to be the pancreatic enzyme that just did its thing, but then the other copy started to change and eventually was selected into what is called an antifreeze protein, a little protein that helped prevent ice crystals from forming in this fish's body cells. And as a result, this fish can live in extremely cold parts of the ocean with no real problem. So very interesting little video clip there. Mutations can also cause changes in genes controlling development. That's kind of a source for some of these larger scale changes that we see over evolutionary time. So an example, heterochrony is where there's changes in the timing of development. And so here we have the skull of a human fetus, and they've kind of superimposed a little grid on it. And here we see the same skull of a chimpanzee, or the same scale, showing a skull of a chimpanzee with that same grid. And then they sort of track, well, what happens to this square, and what happens to this square, and what happens to this square. And you can see that, you know, certain squares change at different rates between the human and the chimpanzee, and the variation in how each of these regions sort of develop accommodates for some of the anatomical differences between a chimp and a human skull. Another example of this, giraffes. A lot of people are surprised to learn that humans have 7 what are called cervical vertebrae. Those are the vertebrae in your neck. And then giraffes also have, wait for it, 7 cervical vertebrae in their neck. So despite its incredibly long neck, the giraffe has the exact same number of vertebrae as a human. How did that happen? Well, the genes controlling the development of those things changed the growth rate. They mutated in some way such that these just grew longer. And so each of their 7 vertebrae are much, much longer than the vertebrae in a human. So that's another example of that. Another one is changes in the homeobox or HOX genes. And what were those things again? Those were those genes that regulate the pattern of body formation. And so you may recall that there's these HOX genes, and they're aligned on a chromosome in a certain order, and that order controls, that order actually matches the order of the body segments that they control. And so this gene regulates the development of this body segment into this head. And you know, this gene controls the development of this segment into this part of the abdomen. And so you can imagine that if we tinker these genes controlling development, we could lead to some very significant changes. And we actually saw that in our genetics unit. We had fruit flies with extra legs or missing eyes or strange things like that. Here's an example of this. Snakes. So snakes are limbless. They have no arms and legs. And it turns out that in a chicken, this is a chicken embryo here, in purple, we have, this is the area where a particular HOX gene suppresses limb development. So it's basically saying, grow limb here in blue, grow limb here in blue, but don't grow any limbs here in purple. Well, in a snake, those HOX genes are active over a much larger area and basically suppresses limb development along its entire length. And so that little mutation there is why snakes have no arms and legs. Here's another one. There's a HOX gene called UBX, and it also suppresses limb development. And so, you know, centipedes, the ancestor insects kind of resembled a centipede, really, but then genes that suppress its limb development eventually caused it to only have these 6 legs, and that basically was the origin of insects. And then we had all these other groups branch off from there. So we can kind of see lots of examples of this, and note that the changes can result from a mutation in the HOX gene itself, or from a mutation in the genes regulating those HOX genes. So remember, there's the genes that do stuff, and then the genes that regulate what is done. So you can see there's lots of examples of this. So then I'm going to pause there, and then we'll pick up on our last 3 mechanisms in our next screencast.